All right, so we're continuing on with our mechanism of action for antibiotics. So we've talked about antibiotics that target cell walls, targeting the cross-linking of the peptidoglycan. We've talked about targeting the folic acid synthesis pathway. We've mentioned those that block the replication of DNA or RNA. And so now we also want to talk about drugs that target protein synthesis, all right? So proteins are instrumental in just about everything that goes on within the cell. So if you remember, proteins are a really large group of macromolecules that are just at work everywhere within the cell. And enzymes are types of proteins. So enzymes are needed to catalyze different reactions, especially different metabolic reactions. And so if you can't make these enzymes, then the cell is going to die. So we know that prokaryotic ribosomes are different from eukaryotic ribosomes. Okay, so remember, the ribosome is the site of protein synthesis. So this is where proteins are going to be made. So if we kind of backtrack a little, if you remember that in order to make a protein, you go through the process of transcription and translation. So transcription is a process where we're making a copy of a portion of the DNA, a gene that has the information on how to make that protein. So we're copying the information from the DNA in the form of a messenger RNA strand, and that messenger RNA is then read by the ribosome and it assembles a protein. So we have drugs that target either the production of the RNA. So it can, some of these antibiotics can target the RNA polymerase enzyme. So without RNA polymerase, we cannot make a messenger RNA strand, which is our copy of how to make that protein. And so therefore, no protein is going to be produced without the instructions on how to make it. Other antibiotics are going to target the ribosomes. So we've got here the prokaryotic ribosome and the eukaryotic ribosome. And we also have this, a different image, but you've seen the same type of thing in previous chapters when we talked about the differences between prokaryotes and eukaryotes. So we talked about the Svedberg units, and so we got our 50S large subunit of our ribosome and our 30S small subunit of our ribosome for prokaryotes. In eukaryotes, there's different Svedberg units. So we got the 60S for the large and the 40S for the small. So therefore, because of the difference in the construction of the ribosome between prokaryotes and eukaryotes, we can be very selective at targeting just the prokaryotic ribosomes and doing no damage to eukaryotic ribosomes. So this group includes your streptomycin, your tetracyclines, erythromycin, azithromycin. All right, so these are kind of your Z-Pack, and they target the 50S subunit. Okay. Some of your tetracyclines, they target the 30S subunit. So it's kind of different in how and what they target, which portion of the ribosome. All right, and our last class of antibiotics targets the outer membrane, typically of gram-negative bacteria. So if you remember, gram-negative bacteria have that outer membrane, which has the lipopolysaccharides embedded in it. And these drugs are typically what we call polymyxins. An older name was colistins, but they're actually polymyxin B and colistin are slightly different from each other. But these are molecules that are able to insert themselves into a membrane. So they actually are built a little bit like, if you remember in membrane construction, the lipid, can't think of the name right now, the phospholipids, there we go. The phospholipids that make up the membranes. So they have a hydrophilic region and a hydrophobic region. So the polymyxins have that dual nature, just like the phospholipid molecule. So they're able to insert themselves into a membrane. And when they do that, they kind of disrupt the membrane. And I've got some images here of some recent research from 2002. And it's showing kind of the side of, they're showing the membrane. And I want you to look at this area right here. So they're showing kind of what the membrane looks like before treatment with an antibiotic and then after treatment with an antibiotic. And it looks like a little bit like a beehive after the antibiotic comes in. So startling difference in the look of the cytoplasm before and after the antibiotic. So the polymyxins kind of interact with the lipopolysaccharides that are in the membrane, and they arrange the lipopolysaccharides into this kind of hexagonal kind of crystalline structure, and it makes the membrane a lot thinner and it makes it stiff as well. And somehow this contributes to cell leakage, so the contents of the cell can leak out, or potential lysis of the cell. So still, ongoing research as to the specific mechanisms of what's going on, but this image here is kind of startling as to what's happening there. So we mainly think of polymyxins as targeting gram-negative bacteria. Their effects on gram-positive bacteria is being researched, so they're not kind of ruling out gram-positive bacteria because there's different acids that these polymyxins could potentially target in gram-positive. But currently they're mainly used with the gram-negatives, and they disrupt the cytoplasmic membrane. So we've spent a lot of time talking about antibiotics, which we said are mainly targeting bacteria. Let's spend a little bit of time talking about other types of antimicrobials for other microorganisms. So there are antiviral drugs as well. So these are much more difficult to develop though, because if you think about it, let's go back and think about what a virus is, right? We said a virus is an infectious agent that is unable to replicate itself. So it needs or requires a host cell in order to produce new viruses. So if the host cell is a human cell and the virus is inside of that human cell, how do you target that virus without affecting the human cell? So it becomes much more difficult to develop these drugs because the target sites aren't as easy. So antivirals that have been developed basically are classified into groups according to the phase of the replication cycle of the virus. So if you remember, we kind of went through that replication cycle. We talked about the attachment to the host cell, penetrating the host cell, how it gets into the host cell, the encoding of the virus, where it releases its DNA or RNA into the host cell, and then it replicates itself, assembles new viruses, and then they are released. So we can look at what phase these antivirals are targeting. So there's inhibition of the virus, keeping it from entering the cell to begin with, and I've got some examples of some antivirals here. You're not responsible for knowing them, but you may have heard of them. We've got inhibition of nucleic acid synthesis, keeping it from producing new DNA or RNA, or inhibiting the assembly or release of the virus. And I want to show you kind of in your D2L site, there is this mechanisms of antimicrobial action here. And you can make it larger by hitting the full screen mode when you go into this site. But this is showing some of these antiviral mechanisms. So target of the virus outside the host, so before it gains access to the host, or target of the virus once it has gotten inside the host cell. So keeping it from entering the host, so entry inhibitor. So here's our virus, here's our host cell. Remember, there's receptors that are on the outside of the host cell, and it has to be able to bind to that host cell. So these antivirals are the ones that are going to block the receptors so that the virus is unable to bind to the host cell. We've got others that block fusion, right? So they might fuse together, especially if there's a membrane around the virus. So if it's an enveloped virus, it might fuse with the membrane of the host cell. And then the other ones are, as we said, once you get in, there's not much really to see in these visuals here. I think the other visuals help you a little bit more, but these are all things that happen once it's inside the host cell. So inhibiting nucleic acid polymerase, so the enzyme that's needed to make the DNA or the RNA, excuse me, protease inhibitors. So as it says here, blocking the protein precursors that are needed for the capsids, integrase inhibitors, keeping the DNA from integrating into the DNA of the host cell, or neuraminidase inhibitors. So remember, these were the spikes on the outside of the virus. They could inhibit that as well. All right, so a lot of different mechanisms there. But once again, kind of following the replication cycle of the virus. All right, so what about fungal infections? So now we're getting to organisms that are eukaryotic, just like our cells. So fungi is once again kind of problematic, because if our cells are eukaryotic and their cells are eukaryotic, they're very similar to each other, and it can once again be hard to find a target to control fungal cells. So we have to look and see what's different. All right, so we need to look at these fungal properties. So here we've got a table that talks about animals, which we fall into that group, versus fungi. So once again, the presence of a cell wall is important. Now this cell wall is very different than that of bacterial cell walls. So there's no peptidoglycan that's present in the cell wall of fungi, and so therefore our antibiotics are not going to work a lot of times against fungi because there's no peptidoglycan. So instead they have a molecule that's called chitin, and that's unique to fungi. So we can use that as a target. And so we have these drugs that are called echinocandins that target the chitin. The membrane of the cell in animals, we find that it possesses cholesterol. Cholesterol is important in the construction of animal cell membranes. Fungi do not have cholesterol, but they do have a different sterol, and that's called ergosterol. And that can be targeted using different drugs. So we see macrolides, the alamines, and the azoles. So azoles specifically are very common broad spectrum antimicrobial agents. So typically treating athlete's foot or ringworm includes different types of azoles. So this last bit, we have antiprotozoal and antihelminth drugs. So we're kind of in the same boat here, where they're both eukaryotes and they have complex life cycles. So with antiprotozoals, drugs usually that target one stage in the life cycle may be ineffective against another stage. So the antiprotozoals are usually split into anti-malarial drugs and non-malarial drugs. Then for the helminths, we have some that basically starve them to death, or paralyze them